moment that we're in, that our community would be very interested to see how science is being applied by the MWRA to learn more about how COVID is impacting our communities. This evening, we're joined by two senior staff at the Massachusetts Water Resources Authority. We have Steve Rode, the Director of Laboratory Services, and Stephen Estes Margiassi, who is the Director of Planning and Sustainability at MWRA. Combined, they have nearly 60 years of experience between the two of them. I'd note that Steve Rode has been thinking about and helping MWRA plan for pandemics for about 16 years. So this is something that MWRA has been thinking about. So without further ado, I'd like to hand this off to Steve Rode, and looking forward to hearing how this collaboration is going between MWRA, Biobot, and Massachusetts Department of Public Health.
All right, let's see. Share my screen here. Maximize that. Come on, slideshow. All right, so thank you, Patrick, for that introduction. You also, or your staff also gave us some talking points, and I'll put them up here for people to see. I'm not going to answer them all right on this slide, but I will put them here, and I will put them up again at the end, and people can tell us how we did as far as answering the questions that are here. And I would actually narrow this down and make it more general and say, how do we get into this? What does this mean, and how does it work? Do you have any data to show us? Of course, everybody always wants to see that. And what's happening next are sort of the basic questions. So jumping in, excuse me. So analyzing wastewater for both for infectious diseases and drugs of abuse and other things is something that has gotten a lot of interest in the last 10 years. It's been used actually for a much longer time than that. People have monitored for polio in wastewater for decades at this point, and some other infectious diseases as well. Biobot is a company that formed with some MIT students and one of their former professors, and they are a company that is specializing in what they call wastewater epidemiology, so looking at wastewater as a way to inform public health of informations at the population level. They started out working on opioid stuff, and they initially approached us a couple of years ago. They were doing a study of a small neighborhood in Cambridge, and they wanted to expand and came to us looking to see if we had sites that were easily accessible to sample that would give them more of the city of Cambridge than the neighborhood that they were already looking at. We were not able to accommodate them because we weren't able to give them sites that met their needs at that time. Excuse me, but they reached out to us in March and said, you know, we think we can analyze for the SARS-CoV-2 virus, the one that causes COVID-19, in the wastewater, and do you think you could provide us some samples so we can try it out? And we were like, yeah, sure, we'll give you some samples, and we did. And they analyzed those. They got some good results that they liked. They went on and created a pro bono program where they invited utilities around the country. Eventually 400 of them signed up in 42 states, including 9 other ones in Massachusetts besides us, to have samples analyzed for the virus. And they shared those results with a lot of utilities and published a couple of different papers. There are other groups around the world that are looking at this. Utilities in Miami, Detroit, Portland, there's a bunch of them now, actually. Those were 3 that were that we knew of when I wrote this slide, which was for another presentation a couple weeks ago. The state of Utah is doing a whole bunch of this wastewater treatment plants in Utah, and they have a nice dashboard that you can go to and look at the different treatment plants and which, how are the data looking. Paris, the Netherlands, Australia, there are a ton of groups actually that are doing this work at this point. And MWRA has provided samples in addition to Biobot. We provided samples to a group of it's being run out of Stanford by a University of Michigan professor who's on sabbatical there. And then they joined up with another research group from Tufts. We provided samples to them. We provided samples to a group from Northeastern, another MIT startup company called Active Signal, and most recently we've provided samples to UMass group at UMass Amherst that are looking at different aspects of the question of using wastewater as a predictor of COVID. We provided some technical guidance to Biobot on 2 papers that they have posted on the internet at this point. We were asked to co-author the second one, and there's a third one that's being worked on at this point. We've been asked to co-author that one as well. Other groups have been doing this. This is a study that was done by a research group at Yale on the city of New Haven sewer system. And basically the signal in red is done. They were working on the sludge rather than the wastewater itself. But the signal, they showed the signal preceding the increase in confirmed cases by about 7 days, increase to a peak, dropped off, with the same sort of scale. Obviously, this is mathematically smooth data. When I get to some real data, you'll see it's a lot noisier than that. So here's another example from Paris, and you can see the red line is the sewage signal from the city of Paris. The purplish line down at the bottom, blue purplish, is actually deaths, and you can see that it again precedes it by about 7 days, the increases. The study was too early, published too early to see if it actually went down in the same way, but it's just another example of this technique being used in different parts of around the world. The Water Research Foundation held a summit at the end of April, and they came up with what's this table of what is the technique useful for at this point, and what needs more work. And so the 4 different cases that they looked at, it's ready for prime time at this point, or almost prime time perhaps, for tracking trends and perhaps the impact of interventions. How does that change the amount of virus that's out in the community? The thought at the time was, and that was, April was still pretty early, that it was good for increasing, that people weren't so certain that it would decrease at the same rate as the cases did. But as you saw from the example that I showed from Yale and some other examples I'm going to show, we are seeing that it does tend to decrease at a similar rate as the number of new cases. Assessing the community level of infection is, another technique or another idea that this could be used for an identification of areas of concern within the community. The far right column is how feasible is the technology for that right now. Assessing the risk to workers in our workers in the wastewater system, and also anybody else in the community who may be exposed to raw sewage, still needs some work. That's why it's a plus minus. But that was one of the cases. And then source tracking of the virus, and I mentioned that we had submitted some samples to a group at Northeastern. They are actually trying to sequence the virus from the wastewater and see if they can come up with the range of different viral signatures that are in the community as a way of assessing how is the virus spreading from person to person within the community. The feeling at the time was that that is a technique that probably needs a lot of work. We gave samples to Northeastern for a number of weeks in March, April, and into May. And then they said, okay, we have enough samples, but we haven't seen any results from them at this point. So perhaps they're learning that it is something that still needs a lot of work. The one thing that everybody wants to be able to do is to say, how many people are infected from this sewer sample? Can you tell me that? And that's actually a very difficult question to answer. And the reason is that, one of the reasons, among others, is that the shedding by patients, and you can see in this graph here, it's a log scale on the left-hand side. So the fecal shedding ranges from 100 copies per gram at the bottom of the red, all the way up to 100 million copies per gram per person. And so how do you tell the difference between that one person who's shedding at 100 million copies or 1,000 that are shedding, you know, at 100,000 copies per gram? And so that's a difficulty that still needs to be worked out in terms of the research. And but it still appears that we can see useful trends with regards to how many people. We just can't necessarily give you an accurate count on how many people that is that are infected at a given time. So the timeline of the work that we've done with Biobot, I mentioned earlier that they requested samples from us in March. They got back to us within a couple days and said, this is working, we want more. And I said, so what kind of control samples are you doing with this? And they said, well, you know, we're still trying to decide what we're going to do with that. And I said, well, I've got a refrigerator full of samples that go back to January before we believe there were any cases in the area. Perhaps you want to try to analyze some of those older samples and just use those as a negative control to confirm. And they were like, yeah. So we gave them a couple of those. And it wasn't too long before they said, can you give us samples for like every week from those samples you gave us in January until now? And we were like, yeah, doesn't cost us anything or to give samples to people who ask if they are doing a good study, which we believe this was. Concurrently, Biobot was working with Boston Water and Sewer, and they went and looked at, I think it was 8 neighborhoods or 9 neighborhoods within the city of Boston of varying sizes, because they were trying to understand how the size of the population served changed the way that the virus behaved within the sewer system. And the second paper that I mentioned, which we worked on with them, also includes that data from the Boston Water and Sewer Commission, and that's available online at this point. And then starting with the governor's reopening plan on May the 19th, you know, they suggested, well, you know, maybe you should give us samples every day so that we can see what happens as these different phases kick in. And so we did that through July 26th. From May 19th, we gave them samples every single day. And some of those results have already been published, and some others are in that third paper that I mentioned that they're working on. So here's the timeline through May, and it does show that there really wasn't, so that the dots are individual samples. The orange dots are the new cases reported each day within. At this point, there weren't always town level data available, and so what they were using was the daily counts for just all of Middlesex, Norfolk, and Suffolk County. So those are the orange dots are the new cases per day, increasing and then decreasing, increasing through April and then decreasing into May. And then they fit curves to those with a 4-day lag, and found that that it's a pretty good match, similar to the other stuff. And that link at the bottom is that second paper that I mentioned of their data. So if somebody wants to go check that out themselves, that's where you need to look. this is our latest data, and it's, like I said, it's a lot noisier, but you can clearly see, that there was no single signal at the beginning of March. It increased very rapidly as the new cases into the beginning of April peaked at some point in early April, and then started dropping, flattened out. We actually, the lowest signal that we got was in the last week of June, and then it rebounded a little bit for a couple days, which led us to do some additional sampling and some additional testing that's not shown here. And then it kind of flattened out again. And we refer to this as our northern and our southern system. So the map on the right shows you what communities are served, what we consider to be the northern part of the system and the southern part of the system. So everything that we did up through June was basically research that we just provided samples, and Biobot did all of the work. Eric Alm's group at MIT did a lot of the lab testing, and a lot of his grad students did a lot of the interpretation of data. And so in June, we decided that yes, this is a useful technique. Let's set something up to make sure we can keep this going. That was around the time that Biobot decided that they had done enough work on the pro bono program to really perfect their technique, and that they were ready to start charging people. And so we, wanted the data to continue, wanted to be continuous. And so we set up a contract with them to collect samples 3 times per week from each of the 2 Deer Island locations, the north side and the south side. So 6 samples per week through December. We built in a buffer of another 40 samples up from upstream locations if we start to see a real increase, that we can move upstream and see if we can decide if it's a certain part of the community that is where the increase is going, or is it really kind of system-wide. So the 6-month contract was basically set up with a not to exceed amount of $200,000, provides continuity, through December. In this program, we will probably go out to bid towards the end of this contract early enough so that we can do comparison data if Biobot is not in fact the low bidder, because there are other companies out there that are working in this field now. The results are available online at the link that's shown. And so we're back around to the talking points. So the origin study, I think I explained, that we had worked with them, or we have been in touch with them in the past, and we got into it simply because they asked for samples. Powerful tool may be an overstatement at this point in time, but it certainly is a tool that has the benefit of sampling all 2 million people at one time, or 1 million people each for each sample at one time, as opposed to the cost of trying. I mean, it would be impossible to go out and sample every single person in a single day to figure out who's infected and who's not. Talked a little bit about who Biobot are, and so we've transitioned from just simply being a partner that provides samples and a little bit of additional technical expertise on how the wastewater system works to we're a paying client at this point. The mechanics of the study, like I said, 2 sample sites 3 times a week. We collect samples at 4 upstream locations and archive those. And so if we see an increase in the signal, we immediately have samples on the shelf from those upstream locations that we can then submit immediately and try to determine if we can figure out what else we can learn about why there's an increase or where the increase is. I showed some sample data. So what's next in this program is that we need to figure out what the lessons learned are. There's a lot to learn. We did do some baseline sampling at those 4 upstream sites and saw some results that were interesting and surprising. And we're still trying to figure out if that's meaningful at all, or if that's really just the nature of how, you know, certain sewer sheds work versus others. I wouldn't say it's being exported elsewhere, although, like I said, you know, there have been several papers posted on the internet at this point. I would say that there are definitely people elsewhere who have looked at those papers and are using them as in their stuff, but those people are also publishing their own work, and we're learning from them at the same time. The Water Research Foundation has put out requests for bids for 2 different studies at this point to study different aspects of it. A couple of the important questions are, how does the signal decay with time in the sewer and time and travel in the sewer? We have a pretty large system and some pretty long travel times in our system. There's a lot of other utilities or municipalities that have much smaller travel times, and so they're going to see different behavior than we are. And also, is there a single sort of best practices for how to do the analyses? I mentioned all these different groups that are involved in this. They're all doing the testing a little bit differently, although it's all based on the same RNA analysis, polymerase chain analysis of that technique, but how they get there is a little different. How they treat the data at the back end is a little different. The Water Research Foundation would like to come up with a set of best practices that, you know, so that data from Detroit will be comparable to data from Boston, from Miami, and whoever else is doing this kind of study. I think I answered all of the questions that are here, but I'm sure people will have some other ones at this point. And I probably was a little faster than 20 minutes, but.
No, Steve, this is Steve Estes from Margianity. Could you touch on 2 things before we go to Q&A? I think you slipped by it on one of your slides, live virus or something other than live virus that we're looking at, and something on variability of flow.
So we are looking at the genetic residual that people excrete. There has been 1 or 2 studies that have been able to find live virus in feces, and there have been perhaps a half dozen others that have not. And so it's still an open question to what extent, you know, actual infectious virus is being emitted by people through this route. But the genetic signal, the genetic residual of the virus, it most definitely is present in feces, and that's what we're measuring.
Talk a little bit about normalizing for flow. You had a bullet point.
Yeah. So and that's one of the things that different groups are doing a little bit differently. What Biobot is using is a virus that infects peppers, called pepper mild mottling virus, and it doesn't infect people, but people consume it by eating peppers and pepper products like Tabasco sauce, and it actually has a fairly steady concentration within the wastewater system. And so when they do the analyses, they actually do the analysis in triplicate for 2 different signals. So we get back 6 different numbers for them, but they normalize all of them to the concentration that they found in the sample of this pepper virus as sort of a way of standardizing for different dilutions. You know, obviously in our system, we're a combined sewer system. So when it rains, we get a lot of extra water relative to the amount of feces that's in the sewer system. And so they use that as a way of correcting for that.
Well, Steve, that was great, super comprehensive. And Stephen, thank you for adding to make sure that Steve hit on all of our topics. We have a number of questions that have come from the community, and I'll encourage others to ask questions in the Q&A area so that we can pass those on. But of course, we have a very much a river focus here. So we'll start with a question about what really there's a question about the river and sewage. So let's start fundamentally. What can you come back to this question of what kind of evidence there is about how virus degrades in sewer water over time?
Certainly. So like I said, nobody has isolated live infectious virus from wastewater directly at this point in time. Lots of people have been able to find the genetic residual, the broken down RNA. But when they're looking for this, they're only looking for two different pieces of the entire genome of the thing. So even the genetic material can be fairly broken down, and they can still make a detection. It's a really sensitive technique.
So what you're saying is suggesting that there's an encouraging aspect of this where it seems like there's not very much live virus, if any, that can be found in the sewage system.
There is actually a paper that's been published or been posted on the internet. I need to be careful about that term these days. There's a difference between published and posted on the internet at this point in time. But posted on the internet that showed that there was actually significant degradation of the virus within the gut environment before the people even excrete it. And so there's still a question as to how much, if any, live virus. Utah, I mentioned, their program is being run by 3 universities within Utah, Utah State, University of Utah, and Brigham Young. They published a paper based on their preliminary data. They included some holding time data, again, just for the genetic material, and they saw even the genetic material signal degraded pretty rapidly within the sewer system.
Great. So I think that really extends to many in our community who are really focused on combined sewer overflows and the concern that this presents an additional risk to the community. Has MWRA given additional thought to this, or does your team feel quite good that this is not presenting an additional risk to the community?
We are fairly confident that it's not creating an additional risk to the community at this point in time, above and beyond sort of the standard risk that wastewater in general provides when it's released into the environment. Great.
Going on down our list of questions, and we've had more than a dozen questions proposed, which is great. We have somebody who really wants to get into the mechanics of this. How do you collect the raw sewage? How do you collect the samples? And does archiving mean freezing the samples? Does Biobot collect them, or does somebody from the MWRA do it?
So we collect them. We as part of operating the wastewater treatment plant, are required to collect samples at various locations within the treatment plant on a daily basis. And so we're using the exact same technology that we use. We set up auto samplers that collect a small amount of sample every 15 minutes over the course of an entire day. That gives us a fairly large sample, and we pour some off when we test the BOD, and we pour some off when we test the suspended solids, and we now pour some off to be tested for the COVID virus.
Great.
So we're not freezing them. There are a number of groups out there that are freezing their samples. There's definitely a case that if you freeze and thaw and freeze and thaw, that will degrade the viral signal. But at this point in time, Biobot prefers to get samples that are simply chilled for analysis.
Mm-hmm. A member of our community is asking, is there a work in progress toward development of a sensor that could acquire this data live and in a continuous fashion?
There is. It's definitely not ready for prime time yet. So Biobot operates out of a, what would you call it? A technology generator space in Cambridge. One of the other companies that operates out of that space is working on sensor technologies for sensing the virus. And at some point, even later this year, they may give us a strip test that we can just simply dip into the wastewater when we collect the sample that will give us a preliminary result.
Great. Is Biobot still at Greentown Labs?
No, well, so their offices are in Somerville. I'm not sure what the name of the facility is, but they moved the lab into a facility called the Engine, which is on Mass Ave in Cambridge, across from the Middle East.
Ah, great. A local landmark here.
Yeah.
Is there, are the results that are collected from a separated sewer system better or have less noise than data collected in the combined sewer systems, like portions of the MWRA?
So, I mentioned the corrections that we do with the pepper virus. That correction is not necessarily perfect. And so one would imagine that if you didn't have changes in flow due to rainfall, you might get a steadier signal than we might be seeing.
Great. Okay. We have a question from Jane Capizzelli here. I'm going to read it to make sure I capture it correctly. She asks, so interesting that you have a promising way of associating the samples with different spatial areas in the city. Any insight how this type of coarse spatial surveillance interacts with traditional epidemiology practices for keeping down the curve and contact tracing that's being done by the city or state?
Yep. Well, this is still an emerging technology, but we are sharing the data as quickly as we get it with folks from the Department of Public Health and the State Command Center. And we actually have a telephone call with them tomorrow to sort of look at, are there better ways that we can share the data or better ways that we can use the data going ahead in time. I'll leave it at that. It's still an emerging technology. I don't think we're ever going to be in a position to make decisions to say we can stop wearing masks because we aren't seeing anything in the sewer system. It's just one of the tools that hopefully will evolve to be one of the tools that's in the toolbox of public health when they recommend those decisions to the politicians who actually make those decisions.
Steve, could you say a little bit about maybe sampling? Some people talking about sampling at nursing homes or campuses or something like that.
Yes, and then there is some of that going on. And I meant to mention earlier on, the state actually has put out a request to solicit a contractor to do other parts of the state outside of the MWRA system. That is out there for people to bid on at this point in time. But one of the companies that we talked to when we were deciding whether we could go with Biobot for this part of this program is actually specializing in smaller facilities. And so they are looking at just that, in individual nursing homes, individual college campuses, and prisons and other types of facilities like that. As part of their selling product, they have also developed a test for testing for the virus on surfaces within a facility. And so, their sort of selling point is, yeah, we'll test the wastewater. If we see an increase in the signal, we'll come into the facility and try to figure out what you need to clean within the facility to fix this problem. Over time, the science is moving away from people getting it from surfaces and more to people getting it from being just simply close to each other. So it remains to be seen how that's going to work out for them. But they're out there, and there are some other companies who have reached out and said, hey, you know, we think we're ready to do this. So we expect we'll get a number of bids when we go out for bid in the fall.
Great.
Patrick, if folks want to see the data rather than having Steve pull it back up, they can just go to MWRA.com, and it's the first link at the top of the page. And the data is being updated every couple of days as we get new data in. So late breaking news as you if you want it.
You know, I'll take my position here to veer away from, we still have a long list of questions to go through, but Massachusetts is seeing an increase in cases now as reported by the Department of Public Health and as reported by others in the New York Times. And you're seeing a modest increase in the RNA signal in your samples. What would you expect, and what conclusions are we drawing from the bump that you're seeing from the MWRA data and the increase in cases? What kind of thinking is going on in discussions?
So you know, I can't speak for public health at this point. One of the things that I do track is how many cases. The state does now put out a weekly report of town by town. And so I tally that up and say how many cases are there within the MWRA system, how many new cases are there within the MWRA system, and what percentage of the new cases within the state is that. That is it's still a heavily weighted sample because a small increase in Boston, you know, outweighs a number of communities in the western part of the state. But the percentage of new cases is down from what it was in late April within our system. So we'll see tomorrow when they put out another update where all these new cases that have been reported this last week were from, but up through last week, you know, the cases were simply in other parts of the state, or the majority of them were in other parts of the state. So there's you go through and read that second paper that the Biobot people posted online there, and it's not just Biobot, it's the MIT research group that they're kind of associated with. They posit an explanation that the signal is actually driven by new cases, that people shed a lot very early on, even before they have symptoms, before they've even got tested, and then it drops off to a much lower level that they perhaps maintain for a number of weeks. But the range is a couple orders of magnitude, and so again, we're in that issue of one new case perhaps being worth 100 or even 1,000 older cases. And so the changes in the signal are driven very much by the new cases, if that's a correct explanation.
I'm still working through our questions here. We have a question from Wendy Seltzer. How common is it for communities in general to store samples, as the MWRA has, that would enable historical analysis of when the virus reached other regions?
I would say it's not very common for smaller wastewater treatment plants. Even the Clinton treatment plant where we run the lab at, they take the samples, they pour off some pieces that come back to our larger lab for testing, and we save those. But the parts that they test at Clinton, they don't have any storage, so they get dumped at the end of the day. And that's more the rule than the exception when you get to smaller treatment plants. Larger treatment plants with large labs like us probably have similar procedures to us, where they're keeping samples for 2 or 3 months before they discard them.
I wanted to go back to some of the earliest questions that we've had to make sure that they were answered, and then just come down through all of them. Julie Ingstern asks, where are the upstream locations that you test when you do see a pulse that you want to know more about?
So we have three large headworks that collect flow from a specific area. And then the fourth is a smaller facility that is actually very specific to two communities that have had high cases over time. And so those are the four locations. It's the three large headworks, and then that one area that services those two communities.
So Chelsea Headworks, is that one of them, or?
Chelsea Headworks is Ward Street Headworks, Columbus Park. The fourth one is the Chelsea Screen House.
Great. And I think Julie asks, and you answered this a little bit, but what are other locations finding that are doing this work that MWRA is doing that has surprised you, or you would highlight, that, or is in parallel with what you're finding?
At this point, so one of the other communities in Massachusetts that is using Biobot to do analysis and is posting data on their website is Nantucket, and they're seeing very low numbers, but also highly variable. Like 1 week that'll be non-detect, the next week they'll have a detect that's, you know, a relatively low number compared to some of what we have seen. But then the next week they'll be back to non-detect, and they're just kind of bouncing back and forth. Now, Nantucket, you know, the entire island is not sewered, so they're not getting the whole island. They've speculated openly that, you know, are the day trippers who come down bringing the signal to them. They collected samples specifically on the 4th of July weekend to test that hypothesis. But they're also only taking a single sample every 2 weeks. And so it's a little bit more difficult to draw a trend and draw conclusions from that.
Great. We have an anonymous attendee who asks two questions. One of them, I think, returns to the hot sauce peppers and coronavirus, whether there's a correlation at all. And they may be referencing the fact that you're using the signal of the pepper as a correction factor, but is there any correlation between pulses of pepper and pulses of COVID would be the question.
So, so we saw when the shutdown, when the stay-at-home order went into effect in March, we saw a drop in our demand for drinking water, and we saw a change in the character of that demand for drinking water that moved out of Boston and into the suburbs. And we saw a similar drop at that time in the wastewater, the dry weather wastewater flow. And then as things have reopened, I haven't looked at the drinking water data, maybe perhaps Steve has, but the wastewater flow seems to have come back, and the pepper virus signal mirrored that to some extent. So it does appear to be a decent indicator of the, if you call it, effective population being served on a day-to-day basis. But other than that, it's also an enveloped virus. And so in terms of degradation within the system, the anticipation is it will behave similarly, and so it makes a good internal standard, but there isn't any specific correlation between that and the coronavirus other than that they have similar properties as viruses.
Okay, great. The same attendee returned to this question of sample collection. Sounds they're familiar with the ISCO and auto sampler. They did ask whether there is some compositing and what the sample size is, and if there is any hold time before you can't really analyze these.
Well, and that's interesting. Nobody's done an official holding time study yet. Yes, We use ISCO samplers and Sigma Auto samplers. They're similar products from two different companies. So these are generally composite samples, although there's been a few days where the composite sample failed and we just took a grab and did that. Biobot actually asks us to give them 3 50 mil centrifuge tubes full of sample. So 150 mils of sample is sufficient for them, and that they analyzed the first one. They're actually freezing the second one and using the third one for QC within the lab if they need it. What was the other half of that question? Composite samples, holding time. Yeah, so there hasn't been a holding time study. When we gave the Biobot March samples, we were giving them initially only 1 sample per week, and at some point they said, we'd like to see if we can fill in the data points on this. And so it was probably the middle of April when we just simply went to the shelf and pulled off samples that we still had archived and poured them off, sent them out, and you saw that you still got a reasonable trend. So over a 6-week period, we didn't lose that much of the signal, enough of the signal to make it more noisy than it needed to be.
Great. I'm really delighted to see we have 2 questions from the wonderful, talented Pallavi Mande from the Charles River Watershed Association, who's joining us this evening. She and Mary White returned to this question about what implications there are for CSO discharges in the Mystic Charles or the Neponset, assuming there is no live virus and just quite a bit of dilution. And added to that from Mary is kayaking safe on the Mystic by Shore Drive, Blessing the Bay? What about the Charles River at Newton and Watertown?
To the best of our knowledge, the standard indicators that would tell you yes or no, can I swim here or kayak here, are a sufficient indication as to whether or not you need to be worried about this particular virus.
Yeah.
Great. I'll note that Pallavi also wanted to ask a question about whether the Watershed Associations could help MWRA with any of the messaging about how our communities and our municipal stormwater department can stay ahead of precautions that communities can take.
Steve, I think this is more your area.
It's a great question. I'm not sure we have an answer tonight, but let's think about it, and maybe we'll get back to Charles River and Mystic and the Neponset with some joint messaging ideas. I think it's, when we can collaborate and present a unified message on some of these topics, it's useful. It's more likely to be heard if it's coming from multiple sources.
Great. Anne-Marie Lambert of Belmont asks, what type of actions are public health or other governmental officials considering as a result of this 7-day warning signal from sewage analysis if it indicates a rise or a fall?
I don't know that I can answer that for them. I think that if it were my decision, I would say, if you think there's a specific area that you've identified as based in the wastewater, then you encourage those people in that neighborhood to get tested, which the state is already doing in the areas where they know that there's been a lot of cases.
Great. Kanan of East Boston asks a question about the July and early August trend, but I think we answered that question about the status of that new incoming data and the trend that we're in right now. Bill Gallagher asks a question about the health of fish and shellfish in the river, trending up, down, or stable. I can answer the river herring portion.
Yeah, that one's yours, Patrick.
Say that it's been a very positive trend for river herring. It was a poor year for all rivers across Massachusetts for river herring. and that has to do with the cohort year, and 3 years ago was not a good year for the fish. But we're hopeful to see a rebound. In general, it's been a very positive trend. And for other fish in the Mystic, we're a really urbanized setting, and we have a long way to go to reduce nutrients and create a more healthy habitat for these fish. So, Bill, I'm really grateful for the question. I'm not sure if our guests have anything to add to the fish question.
Yeah.
2 more questions here. Adriana Silo asks, the wastewater they're using, you're testing, is it the influent wastewater? And do you also test after the secondary effluent, basically before it goes into the outfall tunnel?
And the answer is it is the raw influent. We have not tested further through the treatment plant. However, a number of the other treatment plants that I mentioned have, and they see a large decrease across treatment, and most of them are not able to detect the viral signal after disinfection.
Positive news. And finally, from Wig, and we may have one more late question here, but Wig Zamor, who is a resident of the Watershed and an astute scientist, puts a comment in here. It would be great for you to respond to. Well, SARS-1 was transmitted by a combination of fecal aeration and transfer from building to building via HVAC. The Amoy multifamily housing case from Hong Kong is one of the classics in fecal material and wastewater aeration transmission of a closely related beta coronavirus, and that was published in New England Journal of Medicine. Steve or Stephen, I'm not sure if you've got any comments about how aeration may play a role and whether we should have some concern here.
Steve, you've talked to the researcher, I think.
I have talked to the researcher. I talked to him specifically with regards to safety issues. Well, not necessarily this specific researcher, but one of the researchers who from Hong Kong who looked into this this Amoy Gardens issue, as well as Hong Kong transmission of SARS in general. I asked him specifically. The reason that I talked to him was about any evidence of an increased risk to our workers, to the wastewater workers, because we have areas within our facilities, contained within our facilities, where there is some significant aeration. And he indicated that there were no detected additional cases amongst the wastewater workers, which was a good answer. He also was somewhat dubious about the published claim that it actually transferred from building to building. It's pretty well accepted that within the block G, I believe it was, of the Amoy Gardens, there was spread that is attributed to an improper plumbing practice that was drawing wastewater that somebody on a higher floor had flushed into the bathroom of people below. So that's pretty accepted, but it's a little bit less clear that there was transfer from building to building in that case, or simply that there was what we now have all learned is called community transmission of undetected cases, passing it on to other people within the community.
Great. A question from Srinivas. Can this also be used to detect outbreaks of other diseases in the near future or future?
Yes, absolutely. Anything that that is shed in feces and has an RNA or a DNA signal, probably we will be able to do that. And as I believed I mentioned at the beginning, that people have used this technology in the past for polio and hepatitis outbreaks and some other things. So yes.
Great. Well, we are right up against the clock, and as we expected, this community of people who are interested in rivers and science and appreciative of all the work that MWRA does to deliver such great water to all of our homes and take care of our sewage. We're really appreciative of your work on the forefront to give us this information and make sure we do what we can to reduce the spread of this illness. So to our audience, who numbered nearly 70, thank you for joining us. I'd invite you to get involved with the Mystic River Watershed Association if you're not involved yet. And with that, if there's a way to show our appreciation with thumbs up or hand claps, let's do it. So thank you so much, Stephen and Stephen, for the great work you do.
Thank you. It was great to be here.
Yeah, thanks.
All right. Good night, everybody.